Hi and welcome to the last seminar of the academic year. And I'm very happy to introduce Dr. Catherine Lebel as our speaker today. Catherine is an associate professor of radiology at the University of Calgary and a Canada Research Chair in pediatric imaging. And so Canada Research Chair is a distinguished title that the Canadian government gives a select few university research professors who are also accomplished. And it's a big deal. She leads the Child Brain and Mental Health Program at the Alberta Children's Hospital Research Institute and is a member of the Hotchkiss Brain Institute. And there's some very interesting papers coming out from your group and from this institute, and we all know, you all read them. Catherine received a PhD in biomedical engineering from the University of Alberta and completed her postdoc with Elizabeth Sowell here at UCLA when she was here at UCLA in neurology and pediatrics. Our paths didn't cross back then. I know we talked about this. We may have talked to each other, but maybe not formally introduced. Although I remember sitting in your talk in the Brain Mapping Center, which is the building right behind me on Zoom. So we are coming full circle. So at least you can see that building on Zoom now, even if you're not able to visit. Catherine has written really seminal papers in the field of diffusion imaging in both typically developing children and those with neurodevelopmental disorders, including the fetal alcohol spectrum disorder and learning disabilities. And FASD is something I'm also interested in, and hopefully we hope to have more discussions in that direction sometime soon. Her research is broad in terms of the methods she uses, but also highly focused on brain changes related to cognitive, behavioral, and environmental factors, especially in the prenatal setting. And this is a topic that is very close to us, and many of us here are interested in what you can say. And I should also add that you organized a workshop on trachnometry at OHBM, and I only came to know this at the last minute, which only concluded yesterday. Am I right? So we really appreciate her coming online, despite such a busy schedule, to come and talk to us. So let's welcome Catherine.
Well, thanks very much, Shantanu, for the invitation and for that kind introduction. And you're right, I was thinking a lot about, the last talk I gave at UCLA, which was now, I think, 11 years ago when I interviewed for my postdoc. And so it's nice to be back virtually, even if I can't be back in person, but I have lots of fond memories of my time there. And so it's nice to reconnect. So I'm going to talk today about my work on white matter development in childhood. And I thought I would basically start with my conclusions because, this way, if you drift off or get distracted, you'll get the main points. So the first point, excuse me, I really want to make is that MRI scanning is possible in young children. And in fact, I would say it's critical in young children because this is a really important age range to understand, both in typical and atypical development. So I'm going to show you some of our work in that space. And then I also want to highlight that advanced measures of white matter can give us more specific information about what's going on. So I'm going to show you some of our work in that space in children and adolescents. And then I'm also going to talk again about alterations in kids with prenatal alcohol exposure. And then I'm going to finish up by linking to a different kind of prenatal exposure, which is prenatal stress, and talk about some really new work that we're doing related to the pandemic, which I find very exciting. So structural brain development, we know, proceeds very rapidly in infancy. And, on even on a T1 weighted image, you can see these massive volume increases, for example. And when we look at diffusion measures, you can see really large increases in FA over the first year of life. Almost so large that it kind of looks like things level out after about age 2. But of course, we know that that's not the case. We know from a growing body of literature that FA continues increasing into the 20s, even for a lot of different brain areas. So we can connect the dots or the lines, of course, between this sort of infancy and later childhood and adolescence. And we can obviously infer that there is development occurring between, say, age 2 and age 6. But there really hasn't been a lot of research in that age range characterizing what is actually happening. So, sort of in here, there have been a handful of participants in various studies, but not a lot of work has really focused in on this. And again, I think it's super important to understand what's happening within these kids' brains. But a big part of the reason why research is lacking in that age range is that MRI is challenging. It's challenging to scan these kids. Kids move a huge amount. These are my own kids, and it's hard enough to get, a photo of them. They're always moving. But on top of that, MRI is noisy and unfamiliar for most of these kids. And so they can get scared, fidgety, whatever. All of these combine to mean that they're not necessarily able to stay still for the 30 or 45 minutes that we need to get a good scan. So, they've been ignored largely in the literature, and I'm really interested in this age range. And so we've worked really hard to try to get good imaging data in these young kids. At the Alberta Children's Hospital here in Calgary, we have a dedicated research 3 Tesla scanner. And what we've done is created this rocket ship facade to go over the front of the scanner, just to try to make it a little bit more friendly for the kids. We also have a mock MRI scanner where kids can practice before they do their real scan. And this doesn't have the time pressures associated with the real scanner, but it does have the sounds, and the bed moves in and out. So it does a good job of simulating the real scan. And we've made this look like a rocket ship as well. And then to kind of go along with this, we've developed this astronaut training program that the kids can go through, where they graduate from the mock scanner training as their level 1, and then their level 2 is when they go in the real MRI scanner, and then we say they've finished their training and are now an MRI astronaut. We also have to get creative often with the kids. So I laughed when I came down to the scanner one day and I saw my research coordinator and my PhD student. There's a kid in the scanner, you can sort of see his foot, and his dad is actually lying on top of him, trying to get him to stay still and convince him that he's able to go in. So we get creative, and it takes a lot of patience, but it does pay off in the end. We also have a children's book. This was actually written by one of my research assistants. And it talks about the process of volunteering for a research study, of coming to the children's hospital and riding in the rocket ship. And so we send the ebook version of this, and it's free. I can share the link after, or you can take a screenshot of it now. But we send this to our families ahead of time to help them prepare their kids. And then we have physical copies of the book that we read to the kids, usually before they go in for their scan, just to kind of calm them down and get them familiar with the research team. So using these techniques, we've found them to be quite successful and fun for us. And one of our goals is really to make sure that they're fun for the kids as well, because a real focus of my work is longitudinal imaging. So we want these kids to have a positive experience, and we want them to come back. And we got this spontaneous note from one of our participants that I just think is really cute. So it says, "Dear rocket ship people, thank you for doing rocket ships on me. You probably did it 7 or 8 or 9 times." So she's been back multiple times. "Thank you a lot. It's cool that you let me watch a movie." And so we let them watch a movie too, which is, I admit, a draw for many of them. The other thing we've been doing lately is printing their brains in 3D and giving them a copy of this at a subsequent visit. So we print it out and they can play with it. It's their own brain, about half the size in each dimension. So as I said, we've been finding pretty good success with these techniques. So we've scanned just over 200 kids, and with a total of almost 600 scans now, from just under age 2 to just over age 8. And as I said, we follow many of these kids over many years. We do a lot of different imaging sequences on them, but mostly I'm going to talk about the diffusion MRI today. So it's one of our first sequences. We got 30 directions at B750. And then later in the protocol, if the kids are still happy and calm, we get another set of diffusion images at B2000. We also do anatomical, of course, and passive viewing fMRI. So we let them keep watching their movie while they're in there. When they come in for their scan, we do just a very quick pre-reading language assessment. This is one of the things that we're interested in the lab. But we're really fortunate in that many of the children we recruited for this study came from a different study that recruited women in pregnancy. So many of these kids have very comprehensive prenatal and postnatal histories, which include things like maternal mood, nutrition, substance use, and then infant development, all of those things. So we have quite a rich data set to work with, which is really fantastic. So as I said, I'm going to focus on structural brain development today and really focused on white matter development in particular. This is a T1 weighted image from the same child at 2 and age 4. And although this development is quite a bit less than what we would see from 0 to age 2, you can still see that development is happening that's apparent just to the naked eye. So some small increases in brain volume, of course, but also changes in the white matter volume. You can see, again, just with the naked eye, that it's increasing during these 2 years. And we see that, of course, when we look quantitatively as well. So I'm going to show a lot of plots like this. So just to orient you to what I'm showing, we've got age on the x-axis here. This one has white matter volume here, but you'll see me use other metrics later in the talk. Each of these little gray dots is one scan on one child. And then these red and blue lines are the individual fit trajectories for individual children. So, this line represents one kid. The red are the girls and the blue are the boys. And this black line is the overall fit for the whole group. Okay, so what we can see with white matter volume is, quite large increases. It's almost doubling across this 6-year age range. And it's nonlinear, so it's sort of starting to level off towards the upper end of the age range, but we know it's going to continue for many years. And then looking more specifically at the diffusion measures in the white matter, but this is still a global white matter average, we see increases in FA, and they're moderately large, a lot less than we see in the first couple of years of life again, but still pretty substantial and pretty clear increases of FA with age and very clear decreases of MD with age. You'll note that there's some slight nonlinearity to these curves, more so with the MD, but this is normal. The development's faster at the beginning of the age range. And with MD, we actually see a little bit of a sex difference where the boys tend to have higher higher MD than the girls. Now, I'm really interested in the regional development, and this is what's more novel, is characterizing this early childhood development in more detail than we had before when we were just connecting those infant and older child studies. So what we saw when we looked at the sort of major white matter tracts within the brain is we saw 3 different patterns of development, and we grouped the tracts according to which pattern they followed. So there was this one group of tracts that started with relatively high FA values. Here you'll see it's over 0.5 and had certainly FA increases, but these increases were relatively small compared to some of the other tracts. So starting high with relatively slow development, and this covered the corpus callosum as well as the pyramidal tracts. Then we saw a second group of tracts that had similarly slow-ish development. Again, clear increases of FA, but less substantial than you'll see coming. But they started with low FA values. Here, just 0.36 compared to 0.52 over there. And this was the uncinate fasciculus and the arcuate fasciculus. And then finally, we saw a third group that really had faster development than the other brain areas. So, here you can see this change across the age range is quite a bit more substantial than we see in these other tracts. And so it's really these tracts, primarily association tracts, a lot of them involved in language, actually, which are undergoing pretty major development across this early childhood age range. And this fits nicely with what we know from other literature in that these tracts this is reflecting that they've already undergone probably quite a bit of development and organization, and they're kind of in the home stretch. They're going to keep developing, but they're winding down. These tracts are the ones that are going to continue developing into the 20s and even into the 30s potentially. So they're the slow and steady sort of tracts. They're developing slowly, but they're going to keep going for a very long time. Whereas these are the ones, as I said, that are undergoing the most sort of major development across this early childhood range. I think one of the really interesting things when looking at these trajectories is the individual variation that we see. Looking at this, we can see that individuals vary at any given age range almost as much as we see changes across the age range. And so I think this is really important to note when we're doing studies. And you can see the value in the longitudinal data when we look at individual trajectories, because there's actually quite a bit less variation within individuals. So I'm going to zoom in or highlight a couple of trajectories here. And what you can see when you look at this is we do get variation within these individual scans, but really this child who starts above the curve stays above the curve and follows the same curve. And this child who starts below stays below, again, following very similar development patterns, even with a little bit of variation over time. So to me, this just showcases how important this longitudinal data is. And you can almost think about this like a growth chart for height and weight. It's maybe less important where you start as long as you're following the typical curve. When we would start to get concerned is if they're sort of dropping off or they're not showing that expected increase over time. And I think that's where we can start to learn something about atypical brains and atypical development is in this trajectory here. So on that note, I apologize that the animation just covered everything up, but I'll read it to you. So I do a lot of work in atypical development, looking at kids with prenatal alcohol exposure. And PA is associated with cognitive and behavioral and neurological problems. And it's also associated with facial dysmorphology in some kids, not in all. And the prevalence of fetal alcohol spectrum disorder, which is the neurodevelopmental disorder associated with prenatal alcohol exposure, is quite high. It's estimated it's about 2 to 5%. In the US, the Canadian estimate is about 4%. So to put it in context, I mean, this is 2.5 times more common than autism, but it's relatively understudied compared to something like autism. So we are starting to learn about brain alterations in prenatal alcohol exposure, but we also have a long way to go. And so what we do see is pretty consistently smaller volumes in the subcortical gray matter, and also in terms of cortical thickness, we see reduced cortical thickness. We also see white matter alterations pretty widespread across the brain. So although alcohol exposure tends to impact some areas more than others, there's really doesn't seem to be any particular area that's spared. And then interestingly, we don't just see these static cross-sectional changes, but we also see changes in development rates. So here, what we're looking at is children with prenatal alcohol exposure in blue and children without alcohol exposure in gray and black. And we see these trajectories in blue that are very different from the typical development trajectories in black. Notably, there seems to be sort of less development and less plasticity in these kids with prenatal alcohol exposure. Oh, and there's my animation that works better. Sorry about that. So anyway, we're starting to learn about these brain alterations in these kids, but very much like in typical kids, young kids with PA have not been widely studied. So we know very little about what's going on in the brains of young children with prenatal alcohol exposure. And this is a really important age range in these kids because it's when behavioral and cognitive problems often become apparent. So at least in Canada and in Alberta, we don't typically diagnose FASD until the kids are 6 or 7, because that's when the problems have already become apparent and they can do a proper assessment to get the diagnosis. But we don't know what's happening in the brains of these kids when they're younger. So we looked at 54 children with prenatal alcohol exposure between 2 and just under 7 years. And we compared these to the typically developing group that I just showed you results from earlier. And so we looked at the major white matter tracts in the brain and then compared between the groups. So what we saw were changes in FA, where the group with prenatal alcohol exposure, who's shown in purple here, had higher FA than the group without alcohol exposure, who's shown in this sort of blue teal color. The genu of the corpus callosum was the only area that survived multiple comparison corrections, but you can see a similar pattern across most of the brain areas as well, where the children with prenatal alcohol exposure had higher FA than the controls. We saw a similar pattern with mean diffusivity. So here we saw lower MD in the kids with prenatal alcohol exposure compared to the controls. This was only significant after correction in the uncinate, but again, we see a pretty similar pattern across brain regions. So first, this was a bit surprising. We expected to see opposite results. We expected lower FA and higher MD, which is what has been very commonly reported in older children. Secondly, I think this points to a sort of global brain effect. Although it was only significant in a couple of areas, we are seeing a similar pattern, again, noticing this widespread impact of this alcohol exposure. So given what we know about diffusion parameters over childhood, namely FA goes up and MD goes down, it seems sort of like what we're seeing here is some kind of premature development in these children with PAE. And this is really what I'm interested in. So I want to know, what do the development trajectories look like? We're starting to collect some longitudinal data on these kids. And this is a plot similar to what I've showed you before. So we have the kids with prenatal alcohol exposure, the group fit in blue, and the individual kids are in blue in the background. And then the unexposed controls, the group fit is in black. And what we see here is higher FA in the PA group at these younger age ranges, but overall less or slower development happening in the brain. And when we look towards the upper end of the age range, sort of around 7 or 8, you can see that these lines cross. So this fits really nicely with the literature in older kids, where we have previously seen lower FA in the PAE group. And you can kind of extend this trajectory out and imagine that you would see lower FA. So I think what we're seeing is this earlier development in the kids with PAE, but then also this earlier plateau. So it's still suboptimal development. It's also similar to what I showed before with cortical volume, in that basically we see less change in the brain of the kids with prenatal alcohol exposure. So here they were flatter for volume compared to the controls, and here the slope of the line is less than we see in controls. So overall indicating probably reduced brain plasticity in these kids, which is interesting in that the effects of PAE are very long lasting and they're not static. They have effects on these development trajectories, and also has some interesting potential implications for interventions. Okay, you've seen these images before. I showed them to you at the beginning, and talked about how we know how FA changes in infancy, and we know how it changes in older children and adolescents. And now I hope I filled in that gap for you. But one of the other gaps in the literature is that the vast majority of studies that are out there have used diffusion tensor parameters, so FA and MD as a sort of bread and butter. And those are great. They really are very sensitive to the types of changes we're looking at. And they revealed a huge amount about how the brain develops. But one of the downsides is that they're not very specific. So while we know the brain is changing, we don't really know what in the white matter necessarily is changing. And there are more techniques starting to emerge now that give a little bit of added specificity to these changes. So that's another sort of aspect to my work, is using these slightly more specific techniques to try to figure out what is actually changing within the white matter. One technique that's still a diffusion technique, but is certainly growing in popularity, is neurite orientation, dispersion, and density imaging, or NODI. And there have been a handful of studies that have used this to look at brain development. And they've generally shown that neurite density index, or NDI, which is a measure of axon packing and myelin, is more sensitive to age-related changes than any of the diffusion metrics. So this is a study where right here, they're plotting the correlations with age across different areas of the white matter. And NDI was very strongly correlated with age. The diffusion metrics all sort of show this maybe moderate correlation with age. And then orientation dispersion index, or ODI, was not significantly correlated with age. We looked at this in some of our own data, saw something very similar, NDI showing really nice increases with age across childhood and adolescence, but no changes in orientation dispersion. So already getting a little bit more specific in that these measures are helping to tell us that it's probably not orientation dispersion or axon coherence that's changing, but rather axon packing and myelin. There are also non-diffusion ways to get at myelin and white matter microstructure. And one of those is magnetization transfer imaging. Now, this hasn't been widely used in development, actually, but there are a handful of studies. And interestingly, what they tend to show, now this is a little bit later into adolescence, they tend to show no changes or even a slight decrease in magnetization transfer ratio over time with age. So suggesting that there's probably not a lot happening in the myelin there. But from histological studies, and this is the Yakovlev and Lacour's paper, there is a strong suggestion that myelination continues even during the second and third decades of life, so into the teens and even into young adulthood, particularly in those association areas in the brain. But of course, histology is not in vivo. It's not looking at changes over time within the same individuals. We need imaging for that. And there are some other imaging techniques that try to get at myelin, one of which is myelin water imaging. And this has been used a fair amount now in young kids, so sort of 0 to 5, showing quite large increases in myelin water fraction with some continuing increases, but not as substantial after the first couple of years of life. But it hasn't really been used later into adolescence. So there's also a variant of magnetization transfer, in homogeneous magnetization transfer, which claims to be a little bit more specific to myelin changes. So we have these techniques now. They just haven't all really been used widely in adolescence. So this is what we were interested in doing, was can we tell if there's ongoing myelination in the adolescent period? And beyond that, can we say something about the various processes that are happening? So we collected a large number of different types of imaging measures. We got diffusion at 2 different p-values, so we could get the NODI measures, NDI and ODI. We did myelin water imaging. We did inhomogeneous magnetization transfer, which gives a measure that's a pseudo-quantitative IHMT, so we call that QIHMT. And then by combining information from the myelin water fraction and the NODI measures, we can get an estimate of G ratio. So these are just all of those parameter maps shown in the same child in our study, and then the variation in these parameters along the cortical spinal tract, again, in the same child. So we have all of these measures in the kids, and we're really trying to understand what is happening in the brains. We looked at 50 healthy, typically developing children from 6 to 15, and about half of them came back 2 years later. So the full age range was 6 to almost 18 years with some longitudinal data in there. And as I mentioned, longitudinal data, of course, is so critical here to really understand what's happening over time. So we saw very expectedly widespread increases of FA, decreases of mean diffusivity. We also saw widespread increases of NDI, neurite density index, pretty much all of the tracts within the brain. And then we also saw some increases in myelin volume fraction. So we had three sort of groups of, three patterns of development here, tracts with changes, increases in NDI, but not in any of the myelin measures. Tracts with increases in NDI and myelin volume fraction, and then tracts with changes in NDI, myelin volume fraction, and decreases of G ratio. And I've shown examples, one example of each of those types of tracts down below. So for example, the right cingulum here had increases in NDI, but no changes in myelin volume or G ratio. The body of the corpus callosum had increases of NDI and myelin volume fraction, but no changes in G ratio. And then the uncinate, and this was just the uncinate bilaterally, that showed changes in all of these metrics, increases of NDI, increases in myelin volume, and decreases in G ratio. So we saw no changes in QIHMT, which is that measure that's supposed to be specific to myelin. But it is also a fairly noisy measure, at least in our data. So it's a bit unclear whether that's a sort of sensitivity issue because of the noise or whether that indicates what's actually happening in the brains here. But the myelin volume fraction suggests that something is changing within the myelin, even across this late childhood, adolescence age range. But on top of that, the really robust changes in NDI suggest that this period is very dominated by changes in axon packing, with perhaps some limited increases maybe in the myelin water specifically, but that axon packing is the dominant process occurring here. So you might have noticed when I showed all of those measure maps, or you just know from understanding some of these measures, that there's a lot of shared variance. They share a lot of contrast, similar similar contrasts, and you can see that this is the splenium shown with the metrics, each of the metrics just plotted across it. You can see a lot of similar patterns across the metrics. And then this is a matrix of correlations between each pair of metrics. And we can see very high correlations, for example, between all of the diffusivities. We can see strong inverse correlations between G ratio and myelin volume fraction. Also some nice strong relationships between FA and NDI, for example. So there's a lot of shared variance here. And when we test all of these measures separately, we can lose a lot of power because we're doing so much correction for multiple comparisons. So we were interested in kind of trying to reduce the dimensionality of this and simplify the problem a little bit. And there was a really nice paper out a couple years ago that did just this. So it did a principal component analysis to reduce these diffusion metrics into two components that they suggest represent complexity of the tissue and restriction of the tissue, with complexity reflecting sort of the orientation of the different fibers and restriction being more of a measure of the myelin and the packing. So we had a lot of diffusion metrics, and then we have also these measures that aren't strictly diffusion, but they're white matter measures. And we put them together into a similar type of PCA. And what we ended up getting were three principal components. The first two very similar and very nicely agree with that previous paper. One reflecting tissue complexity. So you can see a big contribution from orientation dispersion here, really getting at, again, how coherent those axons are. Another component with a lot of diffusion metrics here, this also had a big loading from QIHMT, although that's not shown, but representing myelin and axon packing. And then a third component that had our non-diffusion metrics, the myelin volume fraction and then G ratio, that we think probably reflects axon diameter primarily. So we have these 3 components now instead of 9 different metrics. And we looked at how these changed with age in our sample. Component 1, that's the complexity component, was related to age only in the left arcuate nowhere else in the brain. And then component 2, which was this axon packing myelin component, was related to age in almost all of the tracts. And then component 3, this axon diameter, wasn't significantly related to age in any tract. So this is really showing the same thing that we had found with other data, but in a different way, and really confirming that it's axon packing that's dominating these age-related changes, with probably a smaller contribution from myelin. So this is back to the younger kids again, because in a subset of these kids, we did get 2B values in our diffusion data. And so we were able to compute the NODI parameters. So when we looked at this, we see, as expected, nice, robust increases of NDI across the age range that very much agree with the pattern we saw with FA, where we've got some tracts like the corpus callosum having relatively less development, but starting with really high values. Tracts like the uncinate starting with lower values and showing sort of slow and steady development. And then a lot of these association tracts like the inferior fronto-occipital fasciculus here having a bit more substantial development across the age range. So really nice agreement with our FA showing very similar things. We also were able to look at ODI, that other NODI parameter, which reflects orientation dispersion or axon coherence. And we see very slight but significant increases of ODI in a number of tracts, suggesting that they're actually decreasing in coherence across this age range. We don't see changes of ODI after early childhood. And in late childhood adolescence, this tends to be flat. And then when we looked at our IHMT measure, QIHMT, we do see significant increases again in many of the areas. So more strongly suggesting that there is myelination happening across this age range, but those robust NDI changes also suggest axon packing is changing here too. So I want to come back to some different prenatal alcohol exposure data now. And this is a cross-Canada study that had 4 sites looking at over 100 participants with PAE and about the same number of unexposed controls. And I'm focusing on the DTI data here, of course. And this time we used a structural connectome approach to examining the data. So the brain was divided into 90 regions using the automated anatomical labeling template. And then this connectome matrix was binarized to create 1 matrix for the PAE group and 1 matrix for the control group. We looked at the sort of standard connectome measures, and we saw longer, shortest path length in the PAE group, that's here. We saw lower global efficiency, lower degree centrality and participation coefficient, and increased betweenness centrality. But really all of this pointing to less efficiency in the brains of the PAE group compared to the brains in the controls. Another way to look at this data is to look at connectivity within and among different brain networks. So you can use a standard brain network template, and that's what we did here. We divided the brain into 7 different networks. And then we looked at the structural connectivity between different networks and within different networks and looked at whether that was different in the PAE group compared to the control group. And we saw, again, very widespread decreases in this connectivity. So all of the blue lines indicate decreased connectivity in the PAE group compared to the controls. The bold lines are the ones that survived multiple comparison correction. But again, very widespread changes here, although you'll note perhaps that the sensory motor network really comes up as perhaps the most atypical connected network here, in that it has reduced connections with almost every other network, including itself here. In a different study that I'm not going to go into extensively, but we looked at sensory motor connectivity with functional imaging data. And interestingly, we also saw reduced functional connectivity between the sensory motor and the default mode networks. And so functional and structural data do not always agree, but it was kind of nice here to be able to point to a similar finding with functional connectivity that we were finding with the structural connectivity. So I'm going to spend the last few minutes talking about prenatal psychological distress and presenting what I think is some exciting data we're finding in some infants. Normally, depression and/or anxiety affect about 20% of pregnant individuals, so actually pretty common. And this has implications, of course, for the pregnant person themselves, but also for their children, because prenatal distress, like symptoms of depression and anxiety, are associated with an increased risk of behavior problems in kids, as well as a higher risk of later mental health problems when those kids reach adolescence or adulthood. And there's a growing body of literature also showing that prenatal distress is associated with brain alterations in children. So there are a number of studies, but one of the ones that we did recently was looking at EPDS. This is the Edinburgh Postnatal Depression Scale, so a measure of depression symptoms in the third trimester. And then we looked at brain connectivity in the kids between the amygdala and the frontal lobe. And what we saw was that women with higher symptoms of depression had kids who had higher MD values in this tract, so suggesting weaker or reduced connectivity to some extent. And I want to point out here that for the EPDS, cutoffs for that are consistent with a diagnosis of major depression tend to be around 13. So nearly all of the women in this study weren't depressed. They were experiencing a normal range of depressive symptoms, almost all of which were subclinical threshold, but we still see this association between the maternal depression or depressive symptoms and the children's brain connectivity. And then importantly, along these lines, we actually found that there was a mediating relationship. So the mean diffusivity in this right amygdala frontal pathway mediated the association between depressive symptoms in the third trimester and externalizing behavior in the kids. So really suggesting that these brain alterations are a potential mechanism via which these depressive symptoms impact children's behavior. Now, this was only significant in boys in our study, interestingly enough, but there have been just a small handful of other studies that have shown mediating relationships in girls as well. So lots to still be learned about the sex effects there, I think. So obviously, we all know, that's why I'm lecturing over Zoom, that the COVID pandemic has really massively disrupted people's lives. And women, a lot of studies have shown women have been somewhat disproportionately affected, more so than men in terms of job loss or financial hardship or just general life disruptions. And pregnant individuals especially have also been impacted. Their care has been dramatically changed. And again, because of this extra impact on women. So when the pandemic hit, more than a year ago now, we'd been talking about these effects of prenatal stress on kids' brains for a long time, and it seemed like an important opportunity to actually study what massive stress is going to do to kids' brains, especially in this sort of global manner. You know, everybody's been affected to some extent by the pandemic, but obviously some people have been impacted more than others. So we launched a Canada-wide survey of stress during pregnancy, and we ended up recruiting over 11,000 women. They've come from every province and territory in the country, obviously focused in major cities, but we have very good geographical coverage. And so we're continuing to follow these women through pregnancy, and we're obviously following their babies as well, who are being born and growing. So the first thing that we noticed in our study was that the prenatal distress that these individuals are experiencing is way higher than normal and higher even than we expected. The green rectangles here are meta-analyses of pregnancy for anxiety rates and depression rates. So anxiety rates are about 20%, depression rates about 10% of pregnant individuals. This dot and line is the US population norm for adults, not pregnant, but all adults. So it's just a little bit lower than pregnancy for anxiety, a little bit lower for depression as well. But these dots are the data that we had in our study. So 57% of people in our study had clinically elevated symptoms of anxiety, and 37% had elevated symptoms of depression. So massively high, 3 to 4 times higher than what we would normally expect in pregnancy. And obviously very concerning for these individuals and in terms of the potential long-term impacts on their children. So we wanted to look at the kids' brains and try to understand what's going on. So we've been scanning kids born in Calgary. And so far, we've scanned 51 infants born to people in this pandemic study. All of them are full term and have no genetic, neurological, neurodevelopmental diagnoses. And they were scanned during natural sleep on our research dedicated scanner. So I'm focusing on the DTI results today. We also did fMRI and T1 and T2. And in fact, I'm focusing specifically on this amygdala frontal connectivity, which is what I showed you in the previous data, was that mediator between maternal depression symptoms and child behavior. This is a picture of one of our participants. So we looked at these associations, and what we found was a weak but significant relationship between maternal psychological distress. In this case, this is a composite of the anxiety and depression scores, and then amygdala frontal FA in the kids. So in this case, higher distress was associated with very slightly higher FA in the children. So showing a relationship here, but a bit weak. The really interesting relationship came when we added in social support. So social support, we know, can buffer distress itself during pregnancy, but what we found is that it also seems to buffer the brain effects of that stress. In women who had high social support in pregnancy, there was no significant relationship between their psychological distress and their children's brain structure. But in people who had low social support in pregnancy, there was quite a strong relationship where higher psychological distress predicted higher infant FA in this amygdala frontal tract. So of course, this has long-term implications for child behavior and mental health. It also has some interesting implications for interventions in that social support may be an intervention that's positive, not just for mom, but also for long-term effects in the babies. Now, you'll note that this is higher distress to higher FA. So it's kind of coming back to that premature development that we showed in the young kids with PAE. So I just want to touch a little bit on that before I wrap things up. There's growing literature on early adversity. The literature is focused on postnatal adversity, so things like abuse and neglect, that suggests that this early adversity is associated with premature development in terms of advanced DNA methylation and advanced puberty. This is a graph, for example, of threat exposures versus Tanner puberty stage, and more threats were associated with a more advanced stage of puberty. And there's also studies showing faster brain maturation in kids who've been exposed to more traumatic events. So here, the kids who were exposed to more traumatic, stressful events had a higher proportion of their brain that was adult-like compared to the kids with no traumatic, stressful events in their childhood. So it may be that what we're seeing in this prenatal distress and potentially also in the alcohol exposure is a different form of early adversity. Happens prenatally, but it may be having some sort of similar effects on brain development, and that it's happening a little bit too early. Okay, so to kind of wrap everything up, I've talked about early childhood and how there's rapid brain development during that time with quite substantial regional variation. We've shown increasing myelination and axon packing, as well as some increases in orientation dispersion index across that early childhood age range. In the older children, we found increased axon packing quite substantially, with some limited changes in G ratio and myelin water fraction as well. And then I showed a couple of examples of altered brain structure and brain development in kids with prenatal alcohol exposure, both in the young children where we found higher FA and lower MD, and then in the older children where we found that sort of decreased efficiency and connectivity. And then to wrap things up, I showed you some of this new data we have on prenatal stress and brain connectivity in the infants. So coming back to those kind of key points that I want to make, I want to emphasize again that high quality MRI is possible in young children. It requires patience and creativity, but it is possible, and it is very important to be able to understand what's happening in that age range. Longitudinal data is also really critical for understanding the trajectories of brain changes, not just in childhood, but across the lifespan. And I think it shows us a lot more than the cross-sectional studies. We see extensive white matter development in young kids, but then continuing ongoing changes into adolescence, particularly in axon packing. And then we also see that prenatal alcohol exposure is associated with widespread disruptions to brain networks, as well as altered development trajectories. So wrapping everything up, I want to thank my lab, who has been phenomenal, especially during the pandemic and working from home and persevering through all sorts of setbacks. I've listed the people beside there who contributed to the data I showed today. I want to thank my collaborators. I, of course, want to thank the funding organizations that have made this work possible. And I would like to thank you all for your attention. Thank you.
Great talk, Catherine. And at the beginning, I wish you were in the room, and I wish you would see the audience's reactions. I'm sure when you're showing these beautiful slides about kids undergoing brain scans, and really so much hard work going on behind the scenes, and those of us who actually work with the data at the other end. And we do sometimes appreciate what's going on towards collecting data, but maybe not enough. And so thank you again for reminding us, but also at the same time, reinforcing to us that this is indeed possible. So yeah. So any questions from the audience? I have a couple of them, but maybe we can ask the audience if they have any questions.
I have a quick question. So you made the point that high quality MRI is really important, and I agree. And yes, you have a lot of challenges with these kids. In the one experiment, and you compared the different diffusion metrics, I guess, and which I'm assuming involve different sequences as well. And then your latest experiment, looking at infant distress, you seem to just be doing the one, more standard diffusion acquisition. Can you say something about which acquisition type you think is most important to collect in these developmental cohorts, and which I think also extends to, pathology in adults as well? Can you just say something about that? What do you think?
It's a good question, and I struggle in the lab, and I know everyone struggles with this balance between more types of data and better data for one particular modality. So it's kind of the eternal struggle. With our young kids, because there's been so few imaging studies in that early childhood range, we opted to do more of a breadth of imaging techniques. So we got, one diffusion scan, our T1 weighted, our passive viewing fMRI, and then if they were still doing okay, around the 45-minute mark is where we added the second diffusion scan. So we have only a subset of kids with that data, but we still have it. And that's enabled us to do, obviously, multiple studies, not just in diffusion, but in other modalities as well. Whereas in the older kids, given that there's a lot more sort of standard bread and butter technique research, that's where we opted to do the more novel techniques and really dig in a bit more. In our infants, we're doing standard DTI again. We were really prioritizing quick scans. They don't always sleep for very long. We usually get about 20 minutes out of them. So it was sort of what could we fit into 20 minutes? And that was enough to do a quick DTI, a quick resting state, and a T1 and T2 anatomical. If they stay, we do get a second B-value DTI, but we haven't looked at that data at all yet. So for me, it's a matter, I think it ultimately comes down to what's novel and what's our specific question for that group. And in our young kids, it's been more of a bread and butter, what is happening in the brain. And in our older kids, it's been, let's dig in more detail and find out what processes are happening.
Sorry, thank you. I was on mute. That's great. That makes sense.
So then a quick follow-up to that. When you show these neurite density indices, so NODI, do you really require hardy data for it, or can you get by with like a single shell PDI?
So the original version of it required 2 non-zero B-values. So that was generally something around 1,000 and something around 2,000 or 3,000 even. And that's what we did in our kids. There's a new version of it that makes some extra assumptions and does calculate those parameters from a single shell DTI. We haven't tried that in our data. We've always been using the multi-shell. But it does limit your sample a little bit more, but I think it gives better estimates with the 2 shells. Mm-hmm.
Kind of shifting the questions a little bit. First of all, thank you so much for such a fascinating talk. I really enjoyed it. Do you have a sense as to why we would be seeing higher FA values in the participants with the PAE versus controls to begin with?
So, I think I have been thinking about it as a form of early adversity. And the postnatal literature shows that those types of early adversity kind of force the brain and the body to develop a bit prematurely, sort of shut it down, basically. We don't want this extended period of plasticity and vulnerability. We need to kind of get things over with because this environment's not as hospitable, perhaps. And so I've been thinking of this prenatal exposure as well, perhaps as a form of early adversity that's maybe also causing premature development. But I think the really important part to keep in mind is that the premature development is what we see early. And then what we see later is the lower FA, because those lines sort of cross when we look at the development trajectories. So although we often think of higher FA as a better thing, in these young kids, it's not. It means that they're going to plateau essentially too early and have lower FA later on. So I think it's a maladaptive sort of developmental thing. And I don't think it's necessarily a positive. The other thing to note, especially talking about early adversity, is that in many studies of prenatal alcohol exposure, including our own, a lot of the kids that have alcohol exposure also have early postnatal adversity. So they're far more likely to have experienced abuse or neglect or been a witness to violence than the kids that we have in our control group. So it's possible too that there's some interactions with the postnatal environment. And actually, we've been looking at that in some of our other data as well.
Gotcha, thank you.
So follow-up to that is, oh yeah, so I see Mirel actually has to run, but yeah, thanks for a great talk. How do you interpret FA? So there's been a lot of debate about FA not being a structural integrity marker, and especially for maybe typically developing or healthy subjects or normal subjects. But you could interpret it as some kind of an abnormality when you know the disorder, when you know maybe the focal point of that disorder. In kids with PAE, for instance, how you interpret FA or even NODI, these estimates from NODI?
Yeah, it's a good question. And where I always land is that there's something different. So, you know, even in the absence of a very clear interpretation necessarily of the parameters, we know that something is different in the brain. But what does higher or lower FA mean in the case with prenatal alcohol exposure? I mean, it is very likely a combination of multiple different things. I'm sure there are some myelin at play in there and some axon packing. I don't think it reflects like gross damage to the brain. These kids are, you know, for most intents and purposes, developing relatively normally. I mean, their brains don't tend to have gross abnormalities. They're smaller perhaps, but you know, all the parts are there. They're all connected like they should be. So we're seeing relatively subtle changes in the grand scheme of things. Almost very surprisingly subtle given how substantial the behavior and cognitive impairments are for some of the kids. But I don't think we're seeing like major brain damage. I think we're seeing subtle differences in how that white matter is developing. What specifically about it is developing differently, I'm not necessarily sure. I would, you know, throw out the standard explanations for FA, like I said, about packing, myelination, maybe axon size. Yeah.
I have one more question. So you showed really beautiful plots of tracts and these figures, charts, the development. And I noticed that on these charts, you were averaging out the ROIs. Maybe they're coming from tracts, or maybe they're coming from white matter ROIs and so on. What is the role of the structure of the tractography in all this, you think? Or should we really be looking at averaged values of scalar parameters over these tracts?
Yeah, so it's a really good question. And we've thought about this before, and in some of the work, we've also done some additional kind of a long tract analysis to look at different parts along the tract, which I think is part of what you're getting at, right? Is the tract itself developing differently in one part versus another? And can we assume that it's the same, which is what we do when we average across the whole tract. So we haven't done that in all of the work I showed, but in the work we did look at it, which was actually that PCA analysis that I showed with the 3 components, we weren't finding anything substantially different. So it's not as though there was one part of the tract that was massively related to age and another part that wasn't. I mean, we did see obviously some variation along the tract, but nothing that was easily interpretable or felt like it really kind of added to the story. So in the end, we went with the whole tract averages. I think there's a lot to be said for a long tract analysis. That's what that tractometry course was about that we did yesterday.
That's why I asked that question.
Yeah, I certainly think it's valuable, but you always have to balance it with the issue of multiple comparison correction. And so if you don't have a reason to expect the track to be varying substantially across its length, or if in our case, we're often interested in simply characterizing development across the whole brain. I think sometimes it makes sense to limit your number of comparisons and group like with like, which is what we do when we average across the whole arcuate or whatever. So, if I was doing a very detailed study of the arcuate fasciculus itself, I would probably break it down and look at development in different parts. Yeah. So I think they're both good. There's my hedge your bets kind of answer.
Thanks.
I actually
Any other questions?
Yes, I have a question in regards to the PCA. Sorry if I didn't catch it. What proportion or what percent of the data is representative by those two principal components? Like, is there a rationale for why it's just 2 and no further than that?
So this is a good question, and let's see if I can see. I can probably find it very quickly here. Yeah, okay. So the previous study that was published had 2 components. They only used diffusion metrics in their PCA. We added some non-diffusion metrics, the G ratio, the myelin water imaging, and we ended up with 3 components, which captured 38, 23, and 19% of the variance respectively. So really almost all of it, quite substantial there. So I think it's actually a pretty good representation of what's going on with the data.
Okay, thank you.
Okay, so if there are no questions, then we should thank Catherine for such a nice presentation. And we have actually lots of questions as we are thinking about shapes and other things, but we're going to have separate discussions with you, I guess, later sometime. Sure. I know so. So thanks again, Catherine, for coming online and presenting.
Thanks for having me.
And this is the end of the seminar for this academic year. And stay safe, stay healthy. Bye.